Hello, this is Kyung-shin from Korea Research Institute of Chemical Technology. The topic of my presentation is development of a rapid and precise screening method for bioplastic degrading microorganisms. Plastics have made great contributions to humanity with excellent processability properties and low price. The annual generation of plastic waste worldwide estimates at approximately 150 million tons. Plastic waste are mainly managed by disposal in landfills or by incineration. After all, a massive amount of non-degradable plastics is accumulating and intensifying the destruction of the ecosystems. In order to solve this problem, biodegradable plastics such as PLA, PPS, and PPAT have been developed. The degradation of plastic depends not only on the chemical structure of the polymer, but also on the environmental conditions. However, studies on plastic decomposition mechanism have been limited. In order to determine the biodegradation mechanisms of synthetic polymers and estimate the fate of plastics after use, it is important to discovery of novel microorganisms that are able to degrade plastics. However, plastics, including bioplastics, are generally not soluble in water. These properties make the preparation of plastic-containing screening medium fastidious, time-consuming, and labor-intensive. With this, we proposed a new approach for the efficient screening of plastic-degrading microorganisms by spraying a biodegradable plastic solution dissolved in an appropriate organic solvent on agar plate. Compared to other preparation methods, our microspray-based method has the advantages of facilitating plate preparation and ensuring that the plastic particles are evenly distributed throughout the plate surface. For the optimization of spraying condition, we tested the solution concentration and spray time. As the concentration of the plastic solution increased, it was sprayed in the form of fibers rather than the spherical beads. In contrast to the spherical beads, the fibers were not strongly attached on the plate and caused nozzle clogging. Therefore, spraying of a relatively low concentration solution in which fibers are rarely produced is suitable for the fabrication. For example, in the case of PLA, less than 1 poise is suitable. In addition, the amount of plastic particles can be controlled through the spray time, and the particles do not clump together, but are accumulated owing to faster solvent evaporation. This increases the surface area to volume ratio of the particles. When sprayed with 1% plastic solution, the particle sizes of PLA, PPS, PPAT, and PHA were found to be approximately 10 to 20 micrometers in diameter. After the plastic microparticle spraying, the transparent surface of the solid agar plate changed to translucent, and it could be readily distinguished with the naked eye. Before screening wild microorganisms from the environment, we evaluated the screening performance of the screening plate using a previously reported PLA degrading strain, Amycoleptopsis orientalis. After incubation, clear zone formation could be observed and clearly confirmed. To discover bioplastic degrading microorganisms from wide environmental samples, activated sludge samples were inoculated into screening plates. After incubation, it was possible to visually detect clear zones due to the bioplastic degrading activities. We also further investigated the biodegradation of plastic film by isolated bacteria. Isolated bacteria was cultivated with plastic films. After that, pits and crevices were observed on the surface of all the retrieved films. In conclusion, we developed a rapid and precise microspray-based method for bioplastic degrading microorganisms. We had successfully isolated plastic degrading bacteria from activated sludge. We believe our method can accelerate the discovery of novel plastic degrading microbes and can be easily applied to future studies on the decomposition of non-degradable plastics. Thank you for your attention.